Good morning. Today we will be discussing the metabolic engineering of Corynebacterium glutamicum to increase amino acid production. Your objective for this segment will be that given the pathways and mechanisms for production and inhibition of lysine synthesis in Corynebacterium glutamicum, you should be able to recommend changes in key components in the pathway to enhance the cellular production of amino acids, not just lysine, without adversely affecting the organism's growth. The simplified pathway for lysine production and the metabolic engineering associated with it is described here. Aspartate normally runs via several chemical compounds in order to be able to produce lysine. Lysine is feedback inhibited by regulating the production of aspartate kinase once it is produced. This feedback inhibition mechanism affects aspartate kinase, the enzyme, that then shuts down the production of lysine once sufficient quantities are produced by the wild type of this organism. In order to prevent this shutdown from occurring and to increase lysine production, we would like to increase the amount of lysine and continue the operation of aspartate kinase. To do this, we would like to remove the feedback inhibition mechanism, and this is achieved by mutating aspartate kinase, screening it in the presence of analogs of lysine, which we will discuss in a different video, and isolating and quantifying the amount of lysine produced by the so-called regulatory mutants. These regulatory mutants have got the feedback inhibition mechanism switched off. This feedback inhibition mechanism normally operates from lysine to aspartate kinase. You will also note that aspartate splits at aspartate semialdehyde in order to produce at least two more amino acids that are shown. These are homoserine and threonine. Homoserine dehydrogenase catalyzes this reaction. In order to prevent the formation of other amino acids, we would then need to deactivate homoserine dehydrogenase metabolically in order to route the entire production towards lysine. This is what would occur if we removed homoserine dehydrogenase from the equation and did not produce the other amino acids. In order to do this, we would then have to metabolically engineer an organism that does not produce homoserine dehydrogenase. This type of mutant, which requires those amino acids, would then be called an auxotrophic mutant. Also, if we wish to create an auxotrophic mutant, we would have to feed homoserine and the associated methionine and threonine to this organism. A better way to do this would be to create a homoserine dehydrogenase leaky mutant which produces homoserine dehydrogenase at low concentrations, and this would then allow homoserine and threonine to be produced, and this would then allow the organism to continue growing without these feedback inhibition mechanisms occurring, thus increasing the production of lysine. There are two other mechanisms shown for feedback inhibition. One is the repression of homoserine dehydrogenase by methionine, which represses the gene for homoserine dehydrogenase, whereas lysine represses the enzyme aspartate kinase, and threonine represses the enzyme aspartate kinase. Threonine also feedback inhibits homoserine dehydrogenase as well. There are some discussion questions I'd like to pose to you on Blackboard. Please log into Blackboard and answer the question that I am posing. If the focus of production is changed from lysine to threonine in the diagram that I showed you, and you can obtain the diagram in your PowerPoints, what pathways would you modify in the diagram? Explain what type of mutants you would create in order to specifically increase your threonine production. And your solutions and participation in the discussion will be moderated and will contribute towards your course mark. Very briefly, the rubric for discussion would look like this, and I will post an example online for you. Thank you very much.